Assalamualaikum and hello everyone. Let's continue our lesson on mutation. In this video, I will cover subtopic 7.3. Subtopic 7.3 is about chromosomal mutation. The learning outcomes are: define chromosomal mutation, state two types of chromosomal mutation, change in chromosomal structure or chromosomal aberration, and change in chromosomal number. Explain changes in chromosomal structure or chromosomal aberration. Explain alteration of chromosome number. State type of the alteration, aneuploidy or euploidy or polyploidy. Explain aneuploidy and state aneuploidy effect on autosomal chromosome and sex chromosome. Chromosomal mutation is change in numbers or structure of chromosome. Chromosomal mutation has two main classes. The first class is known as chromosomal aberration, when the change occurs in structure of the chromosome. If the change occurs in the number of chromosomes, it is known as chromosomal number alteration. Chromosomal aberration is classified into translocation, deletion, inversion, and duplication. For chromosomal number alteration, it classified into euploidy, or also known as polyploidy, and aneuploidy. Euploidy involves the whole set of chromosomes, while aneuploidy involves the individual number of chromosomes. Euploidy has two classes, autopolyploidy and allopolyploidy. Chromosomal aberration is change in the chromosomal structure, which result in abnormalities in the structure of chromosome. For example, this is the original chromosome. The original chromosome has 6 segments. One of the segments is deleted, or one of the segments is duplicated. Chromosomal aberration occurs due to errors in meiosis or mutations that cause breakage of chromosome. While chromosomal alteration is change in chromosome number, and it occurs due to nondisjunction. Nondisjunction occurs when pairs of homologous chromosomes fail to separate during anaphase 1, or sister chromatids fail to separate during anaphase 2 of meiosis due to failure of spindle fiber formation. When nondisjunction occurs during meiosis 1, when homologous chromosomes fail to separate, it will produce the abnormal gamete with the ploidy level n+1 and another abnormal gamete with the ploidy level n-1 instead of the normal gamete with the ploidy level n. Same goes if the nondisjunction occurs during meiosis 2. The abnormal gametes produced with the ploidy level either n+1 or n-1. When two haploid normal gametes fertilize, it will produce diploid zygote, and this zygote is normal. But when a normal gamete fertilized with abnormal gamete with ploidy level n-1, the zygote produced is abnormal because the ploidy level is 2n-1. This condition known as monosomy. When the normal gamete with the ploidy level n fertilized with abnormal gamete with the ploidy level n+1, the zygote produced is abnormal too, because the ploidy level is 2n+1. This condition known as trisomy. Now we look at the type of alteration of chromosome number. There are two types. The first type is aneuploidy, and the second type is euploidy, or also known as polyploidy. Aneuploidy do not involve the whole set of chromosomes. It occurs due to additional or losing one or more individual chromosome. For example, if the individual has lost one chromosome, the body level is 2n-1, and this condition known as monosomy. But if the individual gain one extra chromosome, the body level is 2n+1, and this condition known as trisomy. Next, we proceed to euploidy, or also known as polyploidy. Euploidy involves the whole set of chromosomes. The individual is not diploid. It might triploid, tetraploid, or pentaploid, and so on. This is the karyotype of a normal human. Human cells normally contain 23 pairs of chromosomes, with autosomes that refer to chromosome 1 to chromosome 22, and sex chromosomes that refer to chromosome 23. Now, what happens if the individual lost or gained one chromosome? So, we look at the effect of aneuploidy. If this occurs in autosome, it's known as autosomal abnormalities. The example is monosomy 21 individual. This individual has 45 chromosomes with ploidy level 2n-1 instead of the normal individual with ploidy level 2n, and the number of chromosomes is 46. The second example is trisomy 21, or also known as Down syndrome. Down syndrome individual has ploidy level 2n+1, and the number of chromosomes is 47, which is extra one chromosome compared to the normal individual. And if the aneuploidy occurs in sex chromosome, it's known as sex chromosomal abnormalities. The example is Klinefelter syndrome, which this individual has 47 chromosomes, which it has one extra sex chromosome. So, the genotype of the sex chromosome is XXY. And the second example is Turner syndrome. And this Turner syndrome has 45 chromosomes, and the genotype of the sex chromosome is XO because this individual has only one sex chromosome. Now, we look at the karyotype of monosomy 21 individual. As we can see in this figure, this person only has 45 chromosomes due to the lack of one chromosome 21. Next is karyotype of trisomy 21 individual. From this karyotype, we can see that this person has 47 chromosomes due to the extra of one chromosome 21. Let's proceed with sex chromosomal abnormalities by looking at the karyotype of Klinefelter syndrome individual. From this diagram, this person has 2 X chromosomes and lead to total of 3 sex chromosomes in diploid organism. Therefore, this individual has 47 chromosomes with a ploidy level 2n plus 1. Lastly, we look at the karyotype of Turner syndrome individual. As we can see, this person has one X chromosome only. Therefore, the number of chromosomes is 45 and the ploidy level is 2n-1. Now, we at the end of our lesson. To test your understanding about this lesson, you may try these 2 questions. Thank you.